welcome everyone to attend our bioinformatics and genomics seminar today. And so Dr. Catherine Bernstein from BCH and I will co-host our seminar today. And so I'm Liang Sun, the senior bioinformatics scientist from the BCH. And before I actually, so Dr. Catherine Bernstein will introduce our speaker today. And before I actually pass it to Kat, so I want to introduce some good resource from BCH. So basically, all our seminars were recorded in YouTube. So let me share my screen so I can show how can you actually find this kind of resource. So if you have browsers, and then you can type RC Wiki from your browser. Oops, sorry. And you will go to our research computing wiki page, and then if you scroll down, you will find the digital science education session, and click that link. You will find what the seminar schedules, and then for example, today we have Dr. Michael Wangler. And then on August 4th, we have a lot of speakers who will introduce the PS polygenetic score catalog. Is one of the database for reproducibility and the systematic evaluation. And what the previous talks actually we recorded in YouTube. So if you scroll down, you can find all the videos here. So if you are interested in any of those talk, you can click and watch it later. And with that, I think I will pass it to Kat. So Kat, can you introduce our speaker?
Hi, can everyone hear me? Can you hear me?
Yes.
Yes, okay. So our speaker today is Dr. Michael Wangler, who is a physician, scientist, and pediatrician, board certified in both pediatrics and medical genetics. And he focuses on the undiagnosed diseases network and the functional analysis of gene candidates for undiagnosed patients through the Model Organism Screening Center. He's used Drosophila models, metabolomics, and genomic sequencing, as well as international collaborations across the UDN and Baylor Hopkins Centers for Mendelian Genomics to solve unsolved cases and study rare Mendelian diseases. He has unique expertise in bridging the clinical practice of medical genetics with the mechanistic understanding that comes from studies of genetic models and computational tools. Team science, collaboration, and linking scientists and clinicians are some of his strengths. And his commitment to undiagnosed disease, rare disease, variant analysis, team science, international collaboration, and functional genomics will undoubtedly shine through his presentation today. It will focus on his new genetics in medicine paper, biases in arginine codon usage correlate with genetic disease risk. And with that, I'll turn things over to Dr. Wangler.
Well, thank you so much. And yeah, just you all can hear me fine, right?
Yes.
Well, great. And yeah, thank you for inviting me to talk. I remember when I was invited, it is intimidating for a clinician to be invited to talk to informaticists about an informatics problem. So, but I'll do my best, And we're going to talk about our paper, biases in arginine codon usage and the correlation with genetic disease risk. I was going to give also some background on how kind of our group got interested in this area. And at the end, I'll talk about kind of some of our thoughts of where we want to go. So as Catherine mentioned in the nice introduction, we kind of see two areas that we look at in our lab. And our idea is that rare and particularly Mendelian diseases give us opportunities. So we have interest in patients that have rare single gene disorders, and we'll apply metabolomics, animal models, genetic screens in models like Drosophila, we'll try to discover new molecular mechanisms. And the hope is that those mechanisms actually may be important for, common disorders and other conditions. And then also one of sort of my passions is, solving undiagnosed cases, having patients coming to my clinic, not knowing their diagnosis. It's, it's just something that you really want to find out at the genetic level, what's causing disease. And for this, whole exome and whole genome has been, just revolutionary in the past few years, as everyone knows. So, for rare disease, I always like to center myself a little bit and, just talk about that this, how do we define this? And so, according to the Orphan Drug Act, any disease that affects fewer than 200,000 would be classified as a rare disease. There's a European definition that's slightly different. A disease affecting fewer than 1 in 2,000 individuals. But, always important to remember that it's pretty common to have a rare disease collectively, because all the rare diseases, if you added it all up, you can come up with, maybe 400 million people worldwide. These are kind of estimates, but it just gives you a sense that rare disease is an important problem in and of itself. But I always also feel like rare disease is something that we focus on, kind of exemplified by this one of my favorite quotes from William Harvey, that basically that nature shows us something important in rare disease by sort of going off the beaten path. And in this quote, Harvey's kind of arguing that, medicine itself, practicing medicine itself, that one of the missions should be to investigate the causes of sort of rare disease. So I'm really lucky at Baylor as a junior faculty in that I get to tap into a lot of resources. So this is like maybe shameless plugging for our institution to you guys, but I just want to, kind of let you know how much resources we get to tap into in our lab. So in the clinic I work in, our clinic sees nearly 2,000 patients with supposed Mendelian or rare diseases every year, chromosomal abnormalities, autism, spectrum disorders. And then we have a large clinical laboratory at Baylor Genetics that has performed, over 10,000 clinical exomes, over 1,000 of those are trios. There's, upwards of 75,000 microarray studies. And then there's several research consortia that also separately have a number of exome databases that we can tap into. And one that I'll mention briefly later in the talk, the Centers for Mendelian Genomics, of which Broad MIT is also a site. This is sort of a large-scale genomic study, and the idea behind the CMGs has been to solve the single gene causes of Mendelian disorders, and that has been a productive database for us, and we have patients that are enrolled in that study. And then, as mentioned, I'm also very invested in the Undiagnosed Diseases Network. So Harvard has a tremendous involvement in the UDN as a joint clinical site, as well as the coordinating center. And then at Baylor, we have DNA sequencing, one of the clinical sites, and then I'm involved in the model organism screening center. And there's other centers around, and the UDN is invested in solving sort of the most difficult N of 1 cases, medical mysteries. And finally, before I kind of move on, we work in the Neurological Research Institute, which is a great place to work for this kind of research, because we're dedicated to kind of pediatric basic science in neurological disease. And so it's a great environment for our lab. So I just want to kind of tell you the story a little bit of how we got to the arginine codon usage ideas for the paper, and then I'll go kind of panel by panel in the paper and explain what we were thinking, and then I'll be sure to leave time for questions. So one thing that got us thinking about arginines quite a bit was actually our work in the Undiagnosed Diseases Network itself, and I'll talk about that. And then I want to cover in some depth visceral myopathy, a specific intestinal genetic disease that really hammered home, at least for me, some of the issues that we approached in the arginine paper. And then I'll talk about the paper. So starting out with the Undiagnosed Disease Network. I think you sort of saw with the panel that there's centers all over. By the way, this was the pre-COVID model of collaborative science, where we all met and had in-person meetings 3 times a year. And by the way, this involved a lot of folks that you guys may interact with. The coordinating center is Zach Cohane, Alexa McRae. I really get along really well with Kim LeBlanc and Cecilia Estevez. And then the Harvard clinical site of the UDN is really impressive. There at Boston Children's, Alan Beggs, Ingrid Holm, Joan Stoller. But then there's also clinicians working at MGH, Brigham. So there's many Harvard affiliates involved in this. And the idea that I like about the UDN is that you bring it together clinicians who are working on unsolved cases, genomics, informatics, and model organism researchers who are like literally doing fly work in the lab, and you have them all talking together about solving cases. So the way that study works is the patients apply through the coordinating center that's actually at Harvard, and then they're assigned to one of the clinical sites. And we're involved kind of usually after sequencing with the model organism screening. So we get nominations of variants from sequencing. And we have an informatics platform called Marvel that we use for individual variants to get at not only human genomic data, but also model organism data that's relevant for that gene and that variant. And then we use that along with other tools to prioritize and then assign. And then genes are assigned to originally they were assigned to Drosophila or zebrafish for laboratory modeling of the specific patient's variant. In the phase 2 expansion of the network, there's also a worm core in St. Louis, an additional zebrafish capacity in St. Louis. And the idea is to then sort of use the model organism data to feed back to diagnosis. So this kind of shows the phase 1 progress that we had in the first part of the study. And we sort of, out of 236 variants that were submitted to the model organism screening, we used informatics to sort of come down to 72 genes that we could look at in either fly or zebrafish, where we could study the gene or the variant and help the patient. And what's shown here is the red would be genes that would be considered gene discovery. By that I mean they're not known Mendelian disease genes that are like indexed in the OMIM database. These are genes that would be kind of brand new single gene disorders. Whereas the black are what we call the phenotypic expansion. So it's already in OMIM, there's already a well-described disorder, but there's something unique about the patient's phenotype, the variant, perhaps it represents a new function for that gene. And this is a big team effort, again, involving a number of people at our institution who sort of help with this aspect of the work. And as I mentioned, now we are actually expanded to multiple sites. So where this sort of intersects with our paper is we've had a number of, we gained a lot of experience over that time in trying to use model systems like Drosophila and, genetically express the patient variant, use rescue paradigms. One we call humanization, where we're essentially removing the fly gene and replacing it with the human gene in the same expression using sort of genetic technology in the fly. And out of this, we actually emerged a number of arginine missense substitutions that had interesting functional consequences in the model. And so I'm gonna go to that in a minute. One of the things about the Model Organism Screening Center that's exciting is that we have this direct communication between the clinicians and the model organism researchers. So we have like monthly group calls, collaborative presentations. All the publications are collaborative. Also in rare disease, it's probably obvious to many people, but matchmaking is crucial. So the UDN kind of selects for these N of 1 cases that are the most difficult cases to solve. And often what you need then is to go find others that have very similar clinical presentations with variants in the same gene. So, we sort of use the resources that I mentioned in the beginning of the talk at Baylor to try to leverage that. And then we try to understand the loss of function phenotype in models as a first step. But it's these variant specific studies that help us, in most cases, get to a diagnosis. So one of the arginine substitutions that I got really interested in was an undiagnosed patient shown here, and she had a cerebellar ataxia. And actually what we found is that she had a unique de novo change, arginine 1673 to proline in CACNA1A. So the CACNA1A gene is actually a well-described disease gene, but this was a really interesting variant in the fly in that we actually found degeneration that was pretty specific to this variant. And we saw some features in this patient that were unique for her, even though she had a lot of features reminiscent of the well-described disease conditions for CACNA1A. And so we actually compared her case and the models to another set of variants within our cohort. And that kind of, I just thought that sort of started me thinking about these arginine substitutions within something like a calcium channel, where you have this portion of the channels involved in voltage gating. And then the other one that had an effect on me was a family that was actually enrolled at the Duke clinical site. And this family, their son is named Quinn. And Quinn had a unique de novo arginine substitution in a gene called NACC1. And what was amazing about this was that Quinn enrolled in the UDN, and we had the NACC1 variant sort of nominated. And within a matter of weeks, we actually found 7 other individuals across different genomic databases with the identical de novo change. So 7 independent de novo events. And so this is obviously due to a hypermutable CPG dinucleotide. And we'll get into that later. But this was just a dramatic example for me in that we kind of went from an unsolved patient to a cohort of 7 families, all with the same de novo change. So anyway, the UDN is is nice. It's a large effort, and it allows us to tap into team science to solve cases. It's nice to see model organism labs that interface directly with clinicians. But it started me on this thought about these de novo, particularly these de novo variants, and these several sort of key arginine missense changes, which, you know, as you'll see later in the talk, I mean, there's nothing new here. But what's interesting is when we start looking at the function in the model, we can start to see unique functional differences of these residues compared to maybe other mutations. So that kind of leads us to the next part, which, so that's sort of one line of evidence that led us to get interested in these arginine codons. The next one was sort of more glaring, I'd say, more obvious. It was just shouting at us. And it came from studies on a different rare disease that's called visceral myopathy. So we had a cohort of patients at Baylor that suffer from a condition that you could summarize as smooth muscle in the body failing to contract to one degree or another, with some patients having almost no smooth muscle function. If you think about your smooth muscle, it's not something you think about very much in your daily life. But if you take away the function of your smooth muscle, it's a really terrible phenotype. It's a really hard thing for these families to go through. You depend on your smooth muscle for every bite of food, every time you go to the bathroom, and even the contraction of your pupils are all dependent on smooth muscle. So this shows a couple patients that we have had in our cohort. At the top panel, the point is just that these patients can have history of sort of gastrointestinal motility problems, where you can inject contrast and you should see the contrast material sort of moving through the gut much faster than it actually, or much slower than it should under normal circumstances, where it's sort of retained days and days after the study. We also have patients who've had such long-standing lack of gastrointestinal motility that you can see this kind of picture where these are actually loops of intestine, and they're just way too large, and they're filled with fluid. And it's basically because things are just not going anywhere. And then this actually shows a bladder, and you'll just have to take my word for it that this is a newborn bladder that's entirely too large for age. And so all these are consequences of gastrointestinal and smooth muscle dysmotility. So we actually had a cohort at Baylor that was from the late 90s. Our former chair, Art Baudet, had interestingly made a mouse at Baylor. And that mouse he was interested in acetylcholine receptors, mainly for autism studies. But when they made this particular ChRNA3 mouse, they actually found that the mouse had an enlarged bladder and a failure of the pupil to contract. So they felt that this mouse probably was the mouse model for this condition, visceral myopathy, which is also known as MMIHS or megacystis microcolon-intestinal hypoperistalsis. But what happened in the late 90s was that they gathered many patient DNA samples, and they sequenced the ChRNA3 gene and basically found no mutations. So then those samples kind of sat until later when exome sequencing came around. And that's kind of when I came on and we had a chance to be involved in finding one of the first genes for this condition, which is actin gamma 2. And so we have a couple of papers, the original paper on actin gamma 2, and then a follow-up from this past year. And so we have 53 families, and what you can see is that actin gamma 2 is by far the most important gene in this condition. So we have 53 families, 38 are sporadic with no affected family members. We actually had, because of the history of it, we had some samples that were collected a long time ago that we resequenced, and then we had sample collection going from 2013 until now. And this has all been work through the Centers for Mendelian Genomics that we've been able to do. And kind of showing that we still have some unsolved, even trio exomes and unsolved proband exomes, but we have a lot of solved cases particularly from actin gamma 2 sequencing. So what stands out when you look at the pathogenic variants in our cohort is that 20 of the 53 probands have an actin gamma 2 de novo mutation. And when you look carefully, there's 5 major arginine residues in this gene. And those are the sites of all the recurrent events. All the other events in the gene that are pathogenic are only seen in one single individual. They're novel variants, but every recurrent variant occurs in this in one of these 5 unique arginine residues. And moreover, if you kind of look at our overall diagnostic rate in that cohort and the number that we diagnosed and the number undiagnosed, the mutation types are, the arginine substitutions outstrip all the other causes of disease in our cohort. So these 5 arginines are really, key. You could just sequence these 5 arginines and solve half your cases in our cohort, which, we thought was pretty impressive. So these, all these things sort of led us to think more about just the basics of arginine substitutions. Very, very basic stuff that Marshall Nirenberg would have, noticed back when the genetic code was being defined. So, there's basically just one thing is that I sort of always had this perception that mutations are pretty random and you can change these amino acids pretty randomly. But if you look and you start to think about a transition event at a CpG dinucleotide, which is by far the most common single nucleotide change that you're going to see, you actually realize that you're in pretty constrained space here. All of your first and second letter CpG positions are arginine residues. You can, you can draw out the consequences of each of these, and they're almost all going to cause missense changes at an arginine residue. You can, of course, find other positions, other codons that have CpGs contained within them. But what's interesting is that a lot of these, because of their position, they may lead to synonymous variation, or you sort of have another set here that we did look at in the paper, by the way, of sort of other amino acids that could be affected by this through specific changes. But what you can see is that if you're thinking about CpG dinucleotides within coding regions, this is very constrained space. And there's nothing new about this. It's just something that we looked very carefully at, and we couldn't help but notice when looking at a gene like actin gamma 2. So, so that kind of leads to the paper, and I'll just summarize the paper in these kind of three areas. So the first question that we're trying to ask is, is there a relationship then between arginine codon usage and particularly these CpG containing codons? Because, by the way, I don't think I sort of glossed over that, but you have, if you think about it, you could also think about this way. You could think that a gene has, 6 options to make arginine, right? It has these hypermutable options, and then it also has these AG containing less mutable options. So that was kind of our thinking, is there a relationship between how a gene makes arginine in those specific positions, its CGN codon usage over the entire gene, and genetic disease risk? So that was one question. We also wanted to know, there's going to be genes that just have high CGN codon usage. What are the characteristics of those genes? And then, are there meaningful differences between specific positions that encode arginine between genes? And I'll kind of cover that at the end. So the first thing we did was we just looked at the ClinVar database. I actually just want to mention, like, this paper, we had, like, it was one of the best kind of peer review experiences I think we've had. The peer reviewers all kind of gave us, we have a bioRxiv version of this paper, and maybe you can take a look and see that it improved quite a bit to the published version. And it was because the peer reviewers really asked for specific things, saying, like, if this is true, then you should see this. And those were things we hadn't quite thought of. So I really appreciated that. And one thing that came up was that, we had to look at ClinVar data. And that was one thing that, peer reviewers really wanted, like, is there an independent confirmatory data set? And that was one thing that we struggled with, is kind of ClinVar seemed to us to be just sort of the best way to get a handle on pathogenic and benign. And you can find other alternatives, of course, but then those partially overlap with ClinVar. So that was the data set that we ended up analyzing pretty deeply. And what we saw is just that if you look at missense, so here on the left is maybe a little bit more of a benign look at the situation, which is that, if you look at pathogenic variants as classified in the ClinVar data, and benign variants that have been classified as benign, arginine actually kind of is the most prevalent amino acid altered in those. And what we're showing here, by the way, is the reference amino acid, right? But then one thing the peer reviewers asked us to do was sort of to normalize that to the size. So we actually looked at the number of the occurrences. So we actually looked at the number of occurrences shown here in panel B. So on the x-axis, we have the number of occurrences of those amino acids in all the canonical transcripts. And that really showed nicely that arginine, even adjusted for how frequently it occurs in a coding region, is a really dominant driver of pathogenic SNVs. So at that point, we were thinking, okay, well, human genes vary in their usage of CGN codons to encode arginine. Some genes are going to naturally have a skewed overrepresentation of those codons. And so, we basically hypothesized that those are the genes that we should look at very carefully, because those are the genes where, for reasons of selection, those codons have been retained, and they're going to be sort of very susceptible to de novo changes that could cause disease. And so what we did here is we compared CGN codon frequency across a set of genes. So here we took the Simons Foundation, autism, And what we're looking here is the category 0, or the syndromic autism spectrum disorder genes. In panel B, we looked at a gene set that we had previously published in our group that, so the reason it's called X-screen is that related to a set of Drosophila genes that we had identified in a neurological screen, but that set of human homologs, it turned out were quite strongly related to single gene disorders. So we sort of had an enriched subset of human single gene disorder genes that we had studied in Drosophila. So sort of a biased subset in panel B. And then in C, we basically looked at OMIM, online Mendelian inheritance in man genes. So, you know, single gene disorders. And what was interesting there is we actually saw a difference between dominant and recessive, which if you consider, that makes sense, because what the hypothesis is that, you know, these are de novo changes that in the heterozygous state could cause disease. So those would be, you would expect those to manifest as dominant disease genes. So then next we kind of looked at whether this correlates to selective constraint. I kind of glossed over that. I sort of said that selection is holding these CGN codons, but we wanted to make sure that that was the case here. So if you look at the PLI score from the NOMAD database or the observed over expected scores, you can see there's a correlation between the CGN codon usage and those levels of constraint. And so then what we wanted to do is we hypothesized that since the GC content at the nucleotide level is sort of known to affect a gene's expression, we wanted to see if the CGN content is related to gene expression. And what was interesting about this was that we're using GTEx data, and we actually saw some tissues showed this and some kind of showed it, or, you know, didn't show it as well. So on the left is just, you know, sort of a more benign check that shows that GC content and the CGN codon frequency are strongly related. But then you can see that if you look at cerebellum and heart, there's a pretty nice relationship there between the expression in those tissues, the level of expression in those tissues, and the frequency of these specific codons across a gene, or the proportion of a gene that uses this codon to encode arginine. But then, interestingly, if you look at liver and skeletal muscle, those relationships don't appear as strong. There does look like maybe there's a relationship there, but it's just certainly not as strong as for brain and heart. And so that was interesting because it may even suggest some tissue-specific expression effects on this sort of set of genes. So finally, that last question was, you know, can you predict in specific positions? And so let me kind of try to explain this figure, unpack it a little bit. So we're starting again from the actin gamma 2, which was the gene I introduced for the visceral myopathy. And in a way it's maybe not the most fair gene to look at because actins are so extremely well conserved across species. But in the human genome, there are 6 actin genes. The actin gamma 1 and actin B are called cytoplasmic actins. And then the C1, ACTA2, ACTA1, and actin gamma 2, these are more tissue-specific actins. So these are highly conserved. Their main difference in the human is where they're expressed. So they effectively make the same protein from all 6 loci, but where they're expressed is what's different. Interestingly, all these are disease genes. So we can actually look at each position. Is there a disease-causing mutation in that position that's known or not? And we can also look, of course, source a database like nomad and just say, is there, you know, an individuals carrying around variants in a population not selected for single gene disorders. And so that's kind of what's shown here, is that we've broken down all of the arginine residues that are encoded in actin. And then in red is shown a CGN codon, whereas blue shows the usage of an AGR codon. And so I think the main point that we saw here is that you can see missense changes in specific sites in nomad, but when you look at sort of ClinVar, you see a non-overlapping set that where there's really interesting positions like the 8th arginine in actin that actually accounts for disease-causing mutations in multiple paralogs. So there's multiple sort of different diseases, but essentially all stemming from the same, you know, 8th arginine in actin encoded by a CG codon. And so, so that was kind of an interesting proof of principle. So I have about 5 minutes left, and then we'll go to questions. So to summarize our paper, the idea is that oh boy, it says my internet connection's unstable. I hope that doesn't have a consequence, but anyway, I'll just keep talking. So, okay, so the first point is that the proportion of CGN codons versus AGR codons actually has surprisingly some capability for Medina disease genes. So again, like the only thing we did was we're just looking in, you know, some of those figures, the only thing we're doing is comparing the 4 CG codons to the 2 AGR codons across all the arginine encoded residues in a coding region. So it's a very simple tool. It's surprising that that has, you know, any predictive capability for disease genes. Genes with more CGN codon usage seem to be more likely, particularly for dominant single gene disorders and in the autism subset, sort of autism genes, if you'll accept that name. And then, so why, you know, what's going on here is one thought, and the argument I want to make is that I think what you're looking at is that there's this awkward balance. You have to think about what's going on at the nucleotide level versus the amino acid level. So at the nucleotide level, some genes need a certain GC content, and they can't maintain that GC content without these CGN codons. There's too much of a proportion of the CG, you know, codons that they require in order to maintain that GC content, which is probably required for expression in certain tissues. But then at the amino acid level, arginine is often important. You need that arginine at that spot. It can't be a different amino acid. So what comes out of that is this sort of awkward balance where you have these genes that retain these hypermutable sites, and those are important genes. And it's important that arginine be at that position, and we're going to see de novo changes in those positions over and over as long as we're seeing patients in clinic. So the hypothesis is really that these CGN genes are good candidates, and we should think about them for allele-specific knockdown therapy because they're going to occur over and over again. And so if you have a gene and you have a site like this is the perfect site to start with for any therapy that would target a specific allele. So, and that's kind of where we're going. Our future directions are, we want to look more carefully at, is there an impact of this codon usage on genotype-phenotype correlation and disease severity? And is there a role for looking at these for gene-specific therapeutic strategies? So I'll just show you these final 2 examples, and then I'll go to questions, okay? So, so when we actually looked back at the original intestinal visceral myopathy cohort, we saw this kind of really interesting relationship. So if we look at disease severity, which we had to define imperfectly, we defined it as individuals that have total parenteral nutrition dependence for a lifetime, transplant, or death from disease. And so we just basically batched patients into 2 groups based on those markers. But what we found is that the actin gamma 2 positive cases account for most of the severe. And then in those, the arginine mutations are disproportionately represented. So those patients are due to arginine substitutions. So we think there may be examples where arginine is actually predictive not only of a disease gene, but also of the disease severity within a disease. And then just gonna mention that another area in our lab, another rare disease that we study is related to peroxisomal disorders and peroxisomal biogenesis disorders. So we've been very interested in these disorders for a long time. These are autosomal recessive disorders, but we've had a couple patients. So we had a recent patient that was a very interesting patient. She came to us because she had been labeled as having cerebral palsy her entire life. And we actually found through exome sequencing and then characterization here, shown here in Drosophila, that her mutation causes peroxisomes to sort of aggregate and fail to divide. And so she actually has a defect in a gene responsible for mitochondrial and peroxisomal fission. And her mutation is not at a CPG, but what was interesting was then we had another patient who we have studies ongoing on his mutation now, and he actually has an R403C. And what's interesting about him is that, sort of as I mentioned, this seems to be the only recurrent mutation that's been seen in multiple patients for this gene, and it occurs at a CPG dinucleotide. And he and the other individuals affected by this mutation appear to have a unique phenotype that's somewhat more severe and different from the other patients. So I kind of had to gloss over that a little bit, but I think the idea here is that this would be a perfect candidate for antisense oligonucleotide therapy. And when we were looking at which mutation should we look at for that, this one really would stand out because we would predict that it would occur over and over again. So anyway, that's all. I just want to acknowledge my lab members. We do a lot of you and stuff, so that this is what we look like. And then of course the great place where I work and all the people involved. It's really more of a team than anything that I really do. So I think I went a couple minutes over, but I think we'll have time for questions.
Yeah, it's really nice talk. Thanks so much. So if anyone have any question, please feel free to send it to Kat or I or me. I can actually unmute you. So maybe I can ask one question first.
Yes.
So it seems like the UDN actually is kind of very useful database. I guess you might also have more things to can dig in, to for the database. So one curious question is that what's the most conserved amino acid residues from your analysis?
Oh yeah, good question. I mean, so since we're doing model organisms, we probably deal with that the most, I'd say that, I don't know how it works for you all. I'd say the sequencing teams rely a lot on things like, gosh, I'm blanking on the tool. There's conservation tools that they'll kind of glance at, but these often end when, sort of zebrafish would be the most distantly related species where people would even look. And the strategy in the, sort of the strategy in worms and flies often might rely on that the amino acid be deeply conserved. I definitely think that we've run into a lot of arginines that are conserved down to worms. But I don't want to tell you that like, oh yeah, it's art, because I don't think we've looked, like we've only really looked at the ones that are submitted to us, and that's a biased subset, because those are variants that the site, sort of has reason to believe that a model organism study would be useful. But definitely, I mean, you can see from the stories that we do run into arginine missense changes quite a bit that are conserved down to worms, including like the CACNA1A. The NACC1 though example, that one's not the gene is not even conserved in invertebrates. So it's sort of a case by case, I guess.
Got it. We have one question from Christy. Christy Murphy.
Hi. I was wondering if you could talk a little bit more about the difference in severity in the condition between those patients who had one of those arginine missense changes versus a patient who did not.
Yeah, so, I mean, what we did in that analysis was, it was fairly bare bones, because we just defined severe based on if you had a like an intestinal transplant, if you had lifelong TPN dependence or death, and and there's other markers of severity that we could have perhaps used, but that seemed to be sort of that's almost like those were clinical markers that people would really, those are clinically dramatic and severe markers, but maybe things like microcolon might have been better. But what I mean, so in that cohort, it really stood out. I just don't know, I think what I want to do though is do that more systematically across diseases. And I don't like, that's tricky to me. I don't know. I mean, I'd be interested if people on the informatics side have ideas about that. I felt comfortable kind of batching the MMIHS patients that way, even though it made obvious assumptions. My problem is, how do you do this across, you'd love to do it across, several thousand single gene disorders, but how do you define disease severity? And it gets into really tricky territory, because families, like even this, when I presented it to the families, I'm very careful, because there's families that, like, I'm calling them mild, and they've gone through real, just terrible, they've just gone through horrible time, but they never had a transplant. So I'm not, so it's kind of like, you have to be really careful how you define severity. And, someone may get an intestinal transplant and live the rest of their life quite well. So, so I would really love a way to sort of define severity, but that's the challenge. I think finding the changes in genomics is easy. It's the clinical part. Like, how could we define severity across many diseases? Sorry, I'm not answering your question at all. I'm just raising, I want you to answer it.
Well, I think I might be the wrong person to answer that, but I think you actually did answer my question, because I was wondering, and I can imagine and appreciate it would be quite difficult to talk about severity, especially because, that can be quite variable, like you said, depending on, especially from the patient perspective, you do have to be careful, using, those words like, mild or moderate with them, just because based on their experience, this might have been, extremely traumatic and severe.
Right, exactly.
Yeah.
And you could maybe go to, there's maybe things, maybe you want to do things that are sort of disease phenotypes that you could quantitate more, maybe through blood pressure or IQ, or I don't, if there was something quantitative, those also have their own problems, and single gene disorders often don't fit well with those quantitative measures. So
Exactly. Yeah.
So
Thank
I mean,
you very much.
I had a plan to maybe just go one by one and just, kind of maybe like the ClinGen model. We just go disease by disease and manually kind of decide, just like we did for MMIHS, this is what would make it severe. This is what would make it less severe. And then try to see if this relationship would hold up. Because right now, obviously, it's just sort of a handful of variants that we've observed look more severe or more interesting.
Right.
Great. We have one more question from Xin Wu.
Hi, Michael. It's an interesting talk. Also love to hear those introduction about and diagnose disease network.
Oh, good.
Yeah, I have two questions about the study that you've done. The first one is that have you compared compare with other amino acids, because there are different parts of amino acid, and it seems that arginine actually has the almost the highest number of permutation, like in the codon table. And so wondering if like based on that, is we can basically understand maybe the number of variants that you found in arginines is probably due to the higher frequency in those genes. So either is that, or it's arginine is more susceptible to variants. So that's one question. The next question is that is the arginine that you observe to hold true for the single gene disorder, or it's more tend to be true to the brain disorder and heart disorder? Because they also show the table that in different GTEx results, yeah, those are the highest, the two tissues that has the highest arginine.
Right. Oh, no, those are both incredible. Thank you. Great questions. So the first question, it's such a cool question because it came up on review, and here's kind of what happened. When we did look, so what we did in response to this, the question is it just the presence of the CG, or is like there's no magic for arginine, right? Is it just the presence of the CG? And so in order to address that, we did a separate analysis that we ended up having a lot of it in the supplement of the paper. But in that analysis, we looked at all of the CG containing codons compared to alternatives. That was an imperfect analysis, though, because you get into these combinations that are more difficult, like if serine to proline. What are the other ways to make serine, and how do you normalize across that? But according to that analysis, your first point is actually spot on. So there's really no magic about arginine itself. It's the CG, it's the CpG dinucleotide. So if you look at those, if you just look at those spots, those are retained the same way that they're retained within an arginine codon in those other codons. But the problem is that the constraint of the space, sort of. So there's only so many ways to make, there's only so many codons that have a CpG. And when you draw it out, arginine has way more ways to sort of go wrong. And so it does seem that arginine, these arginine codons are particularly more responsible for sort of the pathogenic variants in ClinVar, for example. And that's weighted across even their, even how frequent they are within genes or within disease genes. So it's kind of like, there's nothing magic about the arginine other than the constraints that just place it in that where it's the only amino acid. If your codon starts out with a CpG, you're making arginine, and so you're in trouble when you have a transition at either nucleotide. And then your second question is also really great. We suspect that it would relate to disease prevalence, and it kind of relates to the other question. I'd really like to look in more detail at several thousand single gene disorders. But that's kind of where we want to go next. But it is a little more computationally difficult because, like, would we count a disorder that has, sort of cerebellar and liver manifestations, or would we, so we're kind of navigating that. Are we going to do a manual, are we just going to go disease by disease, or is there a better way? Great questions, though. Thanks.
Thank you.
All right. Do we have any more questions? Okay. Thank you so much, Michael.
Thank you.
It's really great talk.
My pleasure.
Yeah.
Thank you.
Yeah. Thanks, everyone. We will see you guys on August 1st.
Thank you.
All right. Bye.
Goodbye. Take care.
Goodbye.